Hey everyone, we'll start on top of the hour, but in the meantime, I shared the lab website and also the science paper and also the free bioarchive version of the paper and the slides on top if you want to, in the meantime, read more about it. The whole group, like 3 people, not the whole group, but part of the whole group that worked on this project will present. So we will have 3 people presenting and discussing, which I think will be really interesting. So yeah, I think it will be a really interesting discussion to have a team here. And yeah, I'm looking forward to it. We'll still have a few minutes to relax. So thanks for coming. And if you think this is interesting, maybe you would like to share the room with people that you know. So thank you. Hi Manas, hi Peter, hi Lubna, and H. Thanks for coming. We'll start on top of the hour, but as I said, there's resources in the chat to check out in the meantime, and also the slides. Yeah, come up, Peter. How are you?
Don't have much to say related to this, but did you have a recording you could share from the room there about the other lady a couple of days ago?
Oh, I thought I sent it to you to email. So did
Oh,
I?
okay. I may have.
So I can resend it
I
again. So how it
yeah, that would be.
works, the link
Yeah,
is only accessible. So when I send it, I'll have to DM you because the link for to download it from the AWS cloud is only active for like 15 minutes or so. So
oh,
I would have to resend it again, and then if you click on the link, it automatically downloads it to your device.
yeah. It
Or
worked. It worked the last time. Great, but so because I thought that that was spectacularly interesting, and my angle was slightly different there, but I think that yeah. So thank
oh,
you
was
very
it a
very much for
different?
that.
Oh, maybe it was a different room. Then send me which room, which
It
date
was the
it
one
was.
about epigenetics. What was her name? Yao
Oh,
or
I see.
something?
Yeah, now I know. Okay, yeah,
Yeah.
just remind me through message because then after the room I will do the same thing again. I'll just share it to your email.
If it's only if it's only valid that short, maybe today is not a good day. I may send you a message tomorrow or something then.
Okay. Yeah.
Okay.
Yeah, I'll do
Thank you
that.
very much. Yeah.
Yeah, sure. No problem. No problem at all. Hi,
So
Matu. Welcome. How are you today? Thank you for coming. To unmute, it's all the way on the bottom right. There's a little microphone symbol. If you press on that, we should be able to hear you. If it doesn't work right away, it could be that it's the first time you're in a room speaking. Sometimes you have to just restart the app and then the second time it works. Can you hear me? Let's see. I wrote in the chat. I know if you can't hear me, it doesn't really help, but let me write you an email. Hi Ganesh, can you hear me? To unmute, it's all the way on the bottom right. There should be a little microphone symbol. And if you press on that, yeah. I can't hear you, but are you talking, Ganesh?
Oh, me?
Yes. Hey, how are you?
Okay. I'm doing good. How are you?
Good, good. You're not by any chance close to Madhu, right? Because I can't, she's not unmuting, and it could be that she's the first time in a room speaking. Sometimes you just have to log back in again, like restart the app, and then the second time it usually works.
Oh, I see. Okay.
So if
Let me
you...
text her.
Oh, great. Perfect. Because if I email her, I'm not sure if she will see it right away. Well, welcome, and thank you so much for coming. This is really wonderful that we'll have a whole team coming to discuss here. I think it will be a really interesting discussion. So,
Yeah, thank you for having us here. This is really interesting to um, discuss this with the community, and it's been kind of collaborative work with me, Madhu, and Simon working. I don't know, since 2016, I suppose.
oh wow, yeah.
Yeah,
So
this is our second paper together.
wonderful. That's a really great collaboration then.
Yes, yes.
And yeah, I will be curious to hear about, how the collaboration came about. But let's start that discussion in a few minutes when everyone
Okay,
is here, when Simon also is here. I added him to the speaker list, so should be
okay.
fine for him that he will be right away up here. Because if he wouldn't, then you would be first in the audience. And for beginners, basically, it's kind of annoying to come
Yeah,
up here. But you've been on Clubhouse, right? So before?
yeah. I've heard other audios. So
Ah, okay, great.
yeah.
But it's the first time you're speaking on Clubhouse?
Yes, this is the
Yes,
first
this is
time
the
that I'm
first
speaking.
time that I'm speaking. Well, it's not really, you don't have to do anything to speak other than having like a profile, to speak
Yeah,
anyway. It's pretty well. Yeah, I don't know, when did you start using Clubhouse as an app? Is it very recent or was it?
after you wrote to us.
Oh, okay, I see.
Yeah.
Yeah, because the app kind of started during COVID, so people were hanging out here basically
Yes,
the whole
yes,
day.
yes. Yeah, I think when it started, I looked at it, and then at that time you needed invites and everything. So
Oh, yeah, I wish I would have known you earlier. I could have sent you
yeah.
an invite. Yeah, it was more like media people and so here, so yeah, you needed an invite. So I felt always weird inviting people to speak because
Hmm.
I would have to send them an invite to their cell phone, and I felt like a scammer. Oh, yeah, talk about your research with us, but I need your cell phone number.
Yeah, yeah.
But most people did, so just a
Okay,
few
give me
people said,
2 minutes. I might have to go troubleshoot mother's phone.
yeah, if
I'll
she
be
can
back.
just restart the app, that would be probably the best way.
Okay.
Or if she's at the university and maybe you're not on the university
She's down the
Wi-Fi,
hall from me. Let me try that.
then switching
Yeah.
to cellular data usually works if
Okay,
some
okay,
universities block the interaction on social media.
okay. Let me go check hers and come back.
Okay. Hi, Simon. How are you? Thank you
Hello,
for coming.
for sure. Thanks for inviting us to give this presentation.
Yeah, wonderful. And it works, so that's perfect. Are you by any chance on a university Wi-Fi or are you using your cellular?
No, I'm on
No.
my, I'm working from home today,
Ah, okay. Yeah, because
so I'm using
Madhu's...
my own personal.
Okay. So I will recommend to
Yeah, I'm not using the serial data. I'm using the Wi-Fi.
But from home, right?
Yes,
Because
yes.
many universities block social media interactions now. So
Oh,
if Matu is on university Wi-Fi, she can be here, but she won't be able to speak or hear us. So
oh, that's weird.
yeah, we had this issue before. So yeah, that's the. It's not every university. It really depends. So um,
It's only through a mobile phone. You don't have any like
there
internet
is a
website that you can.
Yeah, so there is a desktop version app for this. It's
Mm,
Club Deck. It's a little bit more complicated, I feel like, for beginners. It has
I
like
see, I
way
see.
more features, like to add like music, for example, and play it directly, or sounds and different recordings, or analyzing the audience. So it's kind of a little bit less intuitive, I feel like. And then for the recordings, it used to be that you have to have the Clubhouse app to listen to the recordings, but that's not the case anymore, which is good. So I usually share then the link to this recording on the website, but then also we share it on Spotify and YouTube. It kind of
Yeah.
began because younger scientist, she asked me, it would drive up my altmetric score if you would do that, and embed the DOI. So I kind of
Oh, interesting.
started
So,
that, and then it became kind of a thing.
huh,
Yeah,
interesting.
so.
Yeah, I've connected to the Spotify channel to get a sense of how it looks.
Oh, okay, great. Yeah, I was kind of surprised. I didn't even think anyone would go on Spotify to listen, to be honest, because
Mm,
it's not with the slides, right? If you listen to it on Clubhouse, you can still click the link to the slides if the slides are still there. At the clouds spot where you put it during the recording. So I kind of felt, I'm doing this more, to give a little bit something. But then I realized,
mm,
like, 2,000 people or so downloaded the audio files. So I
yeah.
don't know. I was
Well,
so
are the PowerPoint available on Spotify? That's something I didn't notice. I think it's only
no.
the
So
audio recording.
yeah, that's why I thought nobody will care about the spot. It's really just for, the altmetric score of people, and stuff like that. But yeah, people apparently listen to it on Spotify. So.
I see. So the fact that you put on Spotify the DOI increased the altmetric score of the
Yeah,
paper cited.
because it's podcast file, and then YouTube is a video share. So it's like I told usually people, oh, we share it again on Twitter on that day.
Mm,
And she said, that doesn't help me. You shared it already on Twitter.
mm.
If you record this, can't you put this on a podcast or something? And then I made this account. And then, yeah, having a podcast and then YouTube video is a different kind of social media share. So that drives up more the altmetric score than just keeps sharing it on Twitter.
I see, I see, I see.
Mm-hmm. Yeah, it's a whole
Interesting.
science. No, look. Hi, Matu. Thank you so much for coming.
Of
It
course.
works.
Hi,
Perfect.
Katerina. Yeah, I ran into last minute issues with my wireless, so I had to switch. But does it work now? Can you hear me?
Yes,
Yes.
perfect.
Hi,
Yeah, sorry
Sanjay. Hi,
about that.
Ganesh. No, no
We're
problem
missing
at all.
Ganesh now.
I
Oh,
think
I'm
Ganesh
here.
is.
no,
Oh,
no,
yeah, there
he's
he
here.
is.
Everyone's here, so we can start. And people will still keep coming in, but we'll just start with introductions and things like
Sure,
that, and then we'll go from there.
sure.
So, perfect. Welcome everyone to Science Society, and of course, a special welcome to our speaker team today. And before we start, let me just give a brief overview of the speakers we have today. So Kannan is assistant professor and principal investigator at the Department of Neuroscience at the University of Minnesota and the Center for Magnetic Resonance Research. And she is member of the medical discovery team on optical imaging and brain science. And she completed her undergrad degree in biochemistry, and I'm not sure if I'm saying it right, Chennai, India.
That's right.
And her master's in molecular genetics at the University of Leicester. I don't know, UK says things
Yes.
in the UK. And she obtained her PhD in neurophysiology at the Max Planck Institute of Experimental Medicine and George August University in Germany. And during her postdoc, she spent time in the lab of Dr. Mike Hiley at Yale School of Medicine in Connecticut, where she studied mechanism of experience-dependent plasticity at inhibitory synapses of the cortex using slice electrophysiology and optogenetics. And then she joined Dr. Vincent Perrybone's lab for a second postdoc, and there she really started working on emerging genetic engineering and neuroscience. And then we have Ganesh Vazan. He's also a principal investigator and assistant professor in the Department of Neuroscience and a member of the medical discovery team on optical imaging and brain science at the University of Minnesota. And he did his undergrad in electronic sciences at the University of Madras in India. And he did then his master's in nanoscience at the University of Loughborough, UK. And he did his PhD in mechanical engineering at the Max Planck Institute of Iron Research in Germany. And he did later his postdoc also at Dr. Vincent Perrybone lab at Yale, and there he designed and built a high throughput semi-automated platform to engineer fluorescent indicators for cortical and subcortical voltage imaging, and a dual microscope system to simultaneously image odorant response in the dorsal and lateral olfactory bulb. And then we have Simon Hasisa, who is a basic life research scientist at a postdoctoral scholar in biology at Stanford, and he did his master in science at the University of Claude Bernard in Lyon, and his PhD at the École Normale Supérieure de Kashan. I hope I said that right.
Yeah, that's correct.
Perfect. And yeah, it's such an honor having all of you here. And it's really interesting, the team that came basically together to work on this really interesting research. So thank you, first of all.
Yeah. Thank you so much, Katerina. I mean, thank you so much for writing to us. And so this is a fairly collaborative work between two large labs. One was our postdoctoral mentors, Dr. Vincent Pierrebonne, who couldn't be here today. And this was at the Yale School of Medicine and the John B. Pierce Laboratory. And the other large lab that participated was the lab of Professor Mark Schnitzer at Stanford and Howard Hughes, and this is in fact a very large group of people. And today we are the three of us are representing this group. It's going to be Ganesh Vasan, who will be talking about the engineering aspect of the work, the probe engineering and the indicators. And I'll be talking about how we've been using them to acquire insights in the primary visual cortex, and shortly followed by Simon, who will be speaking from Stanford and talking about the applications in hippocampal CA1. So with that, I'll invite Ganesh to take the mic.
If it's okay, we usually do like a very short interview to get kind of an idea about the scientists that are behind the work. So if it's okay, we have usually a question like, how did you, I don't know who would like to answer first. How did you figure out that you want to become a scientist? Was it something you always wanted to do, or was it something that came later, maybe a great teacher or professor or parents that forced you to do it, or a great book or something? If you could give us a little bit of a background, I think it's really interesting for people choosing their career or people that are not in science, how, to get kind of a peek behind the curtain. Thank you.
Sure, sure. Yeah, we could start it in a little bit of a leisurely way. So, I think I got interested when I was in my eighth grade. So, honestly, it was a science exhibition at school where there was one particular exhibit on gene cloning. So that was when Dolly the sheep was cloned, and that kind of caught my attention because there were two words that I didn't know the meaning for. So there was gene and there was cloning, and I had no idea what they meant. So that got me digging into what genetic engineering actually is, and it was extremely insightful to come across the work of like Weymouthian scientists like Professor George Church, and that's when I got pretty much crazy about this field, and I've been pursuing genetic engineering ever since. And my PhD in neurobiology, rather neurophysiology, in Germany further introduced me to the world of neuroscience, and obviously that is something as fascinating or even more fascinating than genetic engineering. So when I got a chance in Vincent's lab to combine the two techniques, that is genetic engineering with neuroscience, I couldn't be happier, and I think this is where my interests currently lie. So yeah, I hope that answers your question.
Yeah, thank you. Thank you so much for giving us that, that insight to, how your path went. I don't know, Ganesh, do you want to go next?
Yeah, sure. I mean, to me, I wanted to be many things probably. So I wanted to be an engineer, I wanted to be a scientist, but I didn't kind of like know what subject that I was interested in. So I tried many things. As you said, my undergrad was in electronic science. Then I was interested in doing nanoscience. And then during my PhD, I did both kind of like material science and mechanical engineering. But it was at that time I also again became interested. I still kind of had an open mind to seeing how can whatever that I've learned in one field can be applied in some other field. So that's when I kind of came across neuroscience. And after moving here to the US, I kind of applied for many neuroscience labs. And until I found one that was brave enough to hire an engineer to work in neuroscience experiments. And Dr. Vincent Pierrepoint's lab gave me that opportunity where I could start up with some engineering projects while at the same time learning neuroscience. So that's where all of this automated high throughput approaches came in. There was a lot of both mechanical and software engineering work involved in that. But along the way, I was able to learn a lot of neuroscience techniques and use my prior knowledge in other engineering fields to apply that to neuroscience to try and answer some questions here.
Yeah, that's interesting that you tried to merge fields. And do you think that's kind of the key to make like major discoveries nowadays? Or it, like I mean, you can merge fields by having a big team of different experts, but do you think we also need people that have kind of a broader approach like
Yeah,
that. Yeah.
yeah, no, I wouldn't recommend that to everybody because it kind of takes a lot of persistence and long periods of confusion, kind of like not knowing where your main interest lies, and it kind of has a lot of challenges. So for a long time, you don't feel like you're a master of anything, right? So, or you understand anything completely. But for persistent people, it might still turn out good. That if they just keep at it, they might finally find what they really want to do. And everything that they've picked up along the way will be helpful in whatever they want to do. But as you said, so some of these can actually be made by real experts in different fields who know what they're doing, so they can come together and try to solve a problem. But there might be some uh, some of the I wouldn't call myself a special one, but to say that you have a little bit of expertise in many fields that you can kind of combine things together. But I wouldn't say that is the key to becoming a scientist. The key to becoming a scientist can still be just specializing in one particular area and becoming a complete expert at it. So there's not just a one way to be a scientist, but this can also be a way. There's still hope.
Thank you for that. And Simon, how about you? Thanks.
Yeah, so it's as Madhu and Ganesh were talking, I was thinking about my own journey, and it's really not linear. And even like if you ask me, if we go back 20 years ago and you ask me, oh, do you think you're going to be a neuroscientist at Stanford 20 years from now? I say no way. There is no way. It's like when I was in middle school and high school, mostly I was mostly better at math, terrible in biology, and pretty low average in physics. And I was terrible in like French and English and stuff like this. And then end of high school, I realized that I had to make a decision about what would be my next step at university or just go working somewhere. So I said, okay, maybe I just go with the path that is the easiest for me, which is math and physics. And I went to engineering schools because that's where I had the highest grades. And then entering my master master degree, and the final 6 months were dedicated in my engineering school for internship, and it was overlapping with something in my personal life where my grandmother was diagnosed with Alzheimer's disease and started bringing a lot of questions about neuroscience, how the brain is working, how come you can like forget your memories and forget the identity of your own grandchildren and stuff like this. So I say, okay, maybe I do my master's thesis at the interface between nanoscience, where I was nanoscale engineer trained, and neuroscience, more flavor of neuropsychiatric and neurodegenerative diseases. And that after my master thesis, everything went well, and my advisor, the two people that soon will become my PhD advisor, offered me a PhD, and I decided to follow this path. And I was mostly working at the nanoscale, between nanotechnology and nanoparticle tracking, and try to understand the neurophysiological basis of autism and schizophrenia. But the nanoscale and single cell resolution studies were not very fulfilling for me, so I decided to go to a more integrated system and work in small animals where the brain is fully wired and integrated with the whole body. And that's where I found this interdisciplinary lab at Stanford from Mark Schnitzer that also had very strong expertise into developing new technologies for understanding fundamental neurobiological questions. And that's it. He asked me if I wanted to work in this project, which was voltage imaging, and a new technology invented back in the day, but fiber optic voltage recording in the 3D behaving animal. I'm going to touch a little bit during the talk. And then that's how I embraced the field of voltage imaging, and I found it. I spent like 6 or 7 years now on this topic, and I had the pleasure to collaborate with Madhu and Ganesh, and it's very fascinating. I think there's tremendous, like a pretty big value for this technology in the coming decades.
Yeah, thank you for that story, and that is also mixed with a very personal story. So, yeah, I think it's always very interesting to learn how paths were made in science or in any other profession, but since we are a science society, I think in science. And yeah, it's wonderful that people found their way and path, and I think it's really a privilege to work in something we are passionate about and actually like. And so, yeah, thank you for sharing those stories with us. And now the stage is yours for the presentation. And everyone, the presentation the link is posted on top, so feel free to follow along with the speakers. And yeah, the stage is yours. Thank you.
Great. Thank you, Katerina. I think probably Ganesh would like to get started.
Yes. Oh, I realize we don't have slide numbers on these. Okay. So I hope everyone's able to open the slides and maybe even start it as a slideshow. So our work started by this trying to answer this question of how do we record what the neurons are doing. So there are, I mean, by saying trying to record what the neurons are doing, I mean one of the primary ways neurons in the brain communicate with each other is through electrical signals, and there are many ways to record this. So if anyone's interested, there's a very nice review from Anne Churchland's lab that was published last year that goes through all of these techniques. And many electrophysiologists, they kind of try to record those activity in single neurons by patching a tiny microelectrode to a neuron. So this is a glass pipette that is supposed to have a very fine tip, and using this you can kind of poke a neuron and try to open its membrane and try to record the potential electric field potential across a neuron's membrane. And whenever neurons, membrane potential changes, you can record these tiny changes in membrane potential. And but the main problem with this is that after this, you're pretty much killing the neurons. You can't record from this neuron, forever, and it's very, very tedious, and it doesn't give you that many neurons that you can record from in a given day. And there are other ways to record electrical activity in neurons. So you can have what is known as extracellular uh, um, field potential recording electrodes, where you kind of insert a microelectrode into the mouse brain or like whatever organism you're trying to record the activity from. And you can record the extracellular uh, field potential from a large number of neurons. And this way it gives you many neurons to kind of like record the activity from. But then again, the problem is this is also a little bit damaging. And if the electrode moves, you won't be recording from the same neuron again. And given that there are many types of neurons in the brain, you won't really know what type of neuron you're recording from. So to solve these problems, you can, try to label your neurons with a fluorescent protein, and then use that somehow to locate which neuron you're recording from. And from that, it kind of branched out into using those fluorescent proteins itself to record the activity from neurons. And in this sense, you have, fluorescent protein indicators that either directly report your electrical activity in neurons, or use some other surrogate, like another ion, for instance. There are indicators that report calcium concentrations in neurons that you can use to interpret activity in neurons. So From this was spawned the two major classes of fluorescent indicators, the calcium and voltage indicators, and they have major differences between them. And um, the main difference is that is what do they report? What do you use as a readout? So in calcium indicators, as the name indicates, you're trying to look at the calcium concentration inside a neuron, and the calcium concentration kind of undergoes a spike inside a neuron whenever there is an action potential. So that is what you use to report, record calcium activity in neurons. On the other hand, for voltage indicators, you have a protein that is sitting directly in the membrane of a neuron. So you can use that to directly record the underlying electrical activity. And because of this, the voltage indicators tend to be much faster in their response compared to the calcium indicator, which rather has a very, very slow dynamics. And you can also have sensitivity to subthreshold events because this calcium spike primarily happens after an action potential is generated. So if you don't have an action potential, you don't have a calcium spike. So you can use a voltage indicator to record these subthreshold activity that is not spike driven. But the main uh, problem for voltage indicators is because of technological limitations of the need to record at very high speeds. The scalability is pretty limited. You can record simultaneously only from a handful of neurons, whereas in calcium recordings, you can run at a normal video rate of like 30 frames per second. So you can record from a large population of neurons. You can record even to about 1,000 neurons in behaving mice. And also for calcium recordings, to go in deeper layers, scientists use the technique called two-photon excitation to record from deeper layers. And the calcium indicators are better suited for this multi-photon imaging technique to record in deeper layers. Whereas again, because of the need for speed, voltage indicators don't have such a technique yet that can be widely adopted by many labs. So to kind of go to what started this story, Mark's lab published, Professor Mark Schmitzer's lab at Stanford, they published a novel class of voltage indicators called the FRET opsin indicators using the opsin protein from the acetable area, and using that, they combined that with a very bright green fluorescent protein called M-neon green. So in this case, the way the voltage indicator works is through the FRET mechanism, where the absorption profile of the opsin that is sitting inside the membrane is modulated by the changes in the membrane potential. And because of the absorption profile changes, during FRET, whatever light that is coming out of the fluorescent protein, a part of it is transferred directly to the opsin through fluorescent resonant energy transfer, or FRET. And the membrane potential modulates the opsin's absorption profile, which in turn modulates the FRET efficiency between the two proteins. So whenever you have a depolarization, you have less light that you can collect from the fluorescent protein because more light is transferred to the opsin through FRET. And this process is near instantaneous. So whenever there is a membrane potential change, within a millisecond, this FRET process is completed, and you can record those underlying electrical activity as very fast fluorescent transients. And you get about a 10% change in fluorescence, whatever you're collecting in your camera, um, during these action potential events. And this was one of the first indicators that was able to demonstrate recording of these electrical activities, not just in vitro, but also in vivo in mice. That was a very, very big feat at that time. At the same time, in the lab of Dr. Vincent Pierrevon at Yale, we were trying to create better voltage indicators through protein engineering, because whenever you combine these two proteins, the voltage indicator and the fluorescent protein, when you put them both together, most often they don't work very well. So you need to perform a lot of protein engineering work to find something that works really good for your setup. And this is quite tedious because you have to create single point mutation in the protein DNA sequence. And as I showed before, you have to perform probably patch clamp electrophysiology, where uh, record the activity from a neuron using a glass micropipette. And doing this one cell at a time for each mutation is very, very tedious. And you won't be able to analyze the entire mutation space if you were doing this manually. So that time when I joined the lab, we were interested in creating some sort of an automated solution to create hundreds of mutations in any given day, so that we can find that one mutation that improves the sensitivity much sooner in within months rather than years, right? So we, along with Dr. Kannan, we created this pipeline of both creating large libraries of mutated DNA from whatever pilot construct we started, and simultaneously perform high throughput fluorescent assays where we express these mutant libraries in some sort of easily cells that you can easily maintain in the lab, and go in with a field electrode, stimulate electrical activity in these cells, and record high-speed video um, in your regular fluorescence microscope. So for this, we created the high throughput platform, as you can see in uh, slide 7, where we came up with kind of a unique spring-loaded electrode design that you can reliably use to, deliver this field stimulation well after well. And because you have to move around and in your microscope and then make sure that your electrode doesn't crush into your microscope or your plate, there was a lot of engineering that was involved in this. And this screening takes about 2 hours to complete. So you have to keep the cells healthy during that time. So we would need to maintain a humid environment. So we were able to achieve all of these and create a platform that was capable of testing um, at least 4 96-well plates, which is about 400 constructs in a single day. And to perform these fluorescence assays, we use what is called an excitable human embryonic kidney cells. These are from the lab of Dr. Adam Cohen at Harvard, and they engineered special types of human embryonic kidney cells that have sodium and potassium channels added in. So these kind of behave like pseudo neurons, where they will have, at a particular density, they will have spontaneous electrical activity. So they will spontaneously depolarize, and at a sufficient density, they will even form these spirals that keep going on and on. So if you're able to play the video on slide 8, you will see these cells expressing ArcLight, one of the earlier kind of like leaders of this voltage indicator group. And so we use these kind of cells to perform our fluorescence assays. And once we express these DNA libraries, we also have another fluorescent tag. So in case your mutation kills your voltage indicator fluorescence, you still need some other way to make sure that you did have expression in your cells, right? So we have a nucleus marker to identify these cells. And in our fluorescence assay, in each well, you move your well plate into your optical path, and then it goes through, let's say, a grid of 16 4x4 fields of view, and identifies the top 4 fields of view to make sure that you have, a good field of view with a large enough number of cells. Because if you randomly go into a well, the chances are high that you might have a region where there aren't that many cells. So you want to kind of look around and find a field of view that has the maximum number of cells. And then you record your high-speed video, and then you identify the cells in each field of view, and then extract their mean fluorescence in each frame. And once you compute the change in fluorescence, both before and after your field stimulation, this percentage of change in fluorescence is what you're interested in. And let's say you start with a probe that has this percentage somewhere around 20%. You're hoping to find a mutation that increases it to 30% or 40%. And one point to note is that all of these voltage indicators work by decrease in fluorescence. That means they start at the resting fluorescence, and then whenever there is a depolarization in neuronal activity, they decrease their fluorescence because of the FRET mechanism that I explained before. And in slide 10, you can see what kind of data we get. So if you have a 96 wells of mutant DNA expressed in these cells, and you go in and apply field stimulation in these wells and recognize the activity, this is the kind of data that you get. And we generate about 1 terabyte of data per plate. And if you're screening thousands of plates, you won't have space to store all of them. So we have to also rapidly analyze all of this data and keep only the fluorescence values that you extract, and we will rapidly delete all of the data, the video data that we collected. So both the acquisition and analysis happens simultaneously. So pretty much in 2 hours, you have the entire result from the plate ready for you. So to see, okay, now we have all of the platform work completed. So now what did we do with this high throughput screening approach? So as I explained before, Mark's lab was had just published Ace-mNeon, where they combined Ace opsin along with an mNeon green fluorescent protein to create a very bright green voltage indicator that was capable of recording single spikes. And we were interested in creating red and even far red voltage indicators at that time, mainly because the longer wavelengths have less scattering and have better penetration to deeper regions of the brain. So we were interested in creating red versions of, well-performing voltage indicators. So as soon as Ace-mNeon came out, we thought, why not replace the mNeon green fluorescent protein with a bright red fluorescent protein, or in this case, mRuby3, that was again published sometime in 2016. So we didn't have success right away, as I mentioned before. So we still had to perform a lot of protein engineering and do high throughput screening to improve the sensitivity to be on par with Ace-mNeon. So in slide 12, if you can see, we were able to record very fast spikes with this new red voltage indicator in both cultured neurons and slices. So this new red voltage indicator we call VARNA, which means color in I suppose Sanskrit. And it expands the voltage activated red neuronal activity monitor, which was just a happy coincidence. So as I said, we were interested in, I mean, what good is a voltage indicator if you can't record spikes in behaving animals in vivo? Because it's all well and good if you can record them in vitro and in slices, but nobody's going to use it if you can't really take them to in vivo recordings in mice. So we continued with our protein engineering approach. And at the end of it, we not only improved Ace-mNeon and VARNAM to create the second generation indicators, Ace-mNeon2 and VARNAM2, we also identified positive variants of these two indicators. And by positive variants, I mean they have increase in their fluorescence response. So instead of being a minus 30% change in fluorescence, now they are plus 30%. And because these are kind of like modular indicators where you can remove the fluorescent protein from one probe and replace it with the red indicator, as I showed before, these mutations are also somewhat transposable. So if you flip the polarity in the green indicator, you can use the same mutations to reverse the polarity in the red indicator. So, these all four indicators have really good kinetics. They are ultra fast. Their response time is for Ace-mNeon2, VARNAM2, and the positive indicators, which we now call positive ACE or PACE, and positive ACE in red or PACER. So all four of them have really good kinetics that you can use to record single spikes in vitro. And so we wanted to test their performance in vivo now, because that's what we were all aiming for. We were not able to do that for many, many years. And that's what all the labs that were developing voltage indicators were aiming for. And so just like Ace-mNeon, Ace-mNeon2 had even better sensitivity for recording spikes in vivo in mice. And previously we were able to record maybe just from 1 or 2 cells. But in this case, because of the improved sensitivity, we were able to record from over 50 cells in a single field of view. And this was a major achievement for us, because now you can start doing some meaningful experiments, because now you can start recording from not just 1 or 2 cells like what electrophysiologists do, but you can start recording from 50, 60 cells uh, in awake behaving mice. So not just the negative indicator, Ace-mNeon2, and in slide 16, if you see, even the positive indicator, PACE, is also capable of recording spikes with superior sensitivity in awake behaving animals. So now this gave us an idea. Now you have 2 indicators, or like 4 indicators. If you look at slide 17, all 4 indicators, you can record spikes in vivo in awake behaving mice. So normally, if you were to record, as I said, if you were to record activity from multiple neuron types, you need to know which type of neuron you're recording your activity from. You would typically combine a green and red indicator together and perform a dual color imaging, right? This is what neuroscientists have done. Even if you're labeling just tissue, not even recording activity, if you want to differentiate what you're imaging, you would usually separate them by the spectral color, spectral wavelength, and you go blue, green, red, infrared. So you kind of differentiate by the color of the fluorescent protein you use to tag whatever you're interested in recording. So in this case, now we have 2 indicators: the negative green Ace-mNeon and the positive green PACE, that can potentially be combined to record activity from 2 cell types, where you can differentiate them based on their polarity. So if you look at slide 18, this shows how it works. So you have a double transgenic animal, and you perform conditional expression of these 2 indicators in a Cree and a flip recombinase mouse, and you can then perform your voltage imaging in a single color channel, just a regular GFP fluorescence microscope. And finally, in analysis, you can identify which direction your voltage transients are. So if they are negative, then you can say this is coming from Ace-mNeon2, and you used Ace-mNeon2 to label a particular cell type. So this spike recording should be coming from this cell type. Same way for PACE. You see more positive spikes in your trace. So you say, okay, this trace should be from, PACE voltage indicator, and you use that to label this particular cell type. So this trace should be coming from this cell type. So in slide 19, if you look at it, if you can play the video, this is what it looks like in practice. So you perform a single channel voltage imaging, and you get about 20 to 30 cells, and you can simply separate these 2 groups of cells based on which direction their spikes are. And we were quite excited to try this. This took a lot of planning, and which cell types we try and all of those things. But we were really, really happy when this finally worked. And we thought, okay, now we have a very, very powerful tool to not just look at activity from single cell types, but actually start performing some local circuit computations in mice as they are doing some sort of behavior. And you will hear a lot more about it from Dr. Kannan and Dr. Aziza when they talk about their behavioral experiments in visual cortex and hippocampus. With that, I'll have Madhu take over from the next slide.
Thank you, Ganesh. I don't know if Katerina has any remarks here or any questions from anyone.
Yeah, let me check in the chat. So far, I think it seems like there are no questions from the audience, I could just comment that it's really impressive. The time resolution you have, and also the this 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 voltage indicators, how well the signals are aligned with the recordings, like the electrophysiology recordings. It's really impressive. And do you, for coming up with these indicators, these genetic indicators, did you beforehand, maybe this could be a question from the audience, did you beforehand, what work did you do to prepare this to make them so efficient and to work so well? Was it modeling maybe before? Did you analyze the structure, or did you just do trial and error basically and then come up with this specific ones?
A bit of both, mainly because you know how these voltage indicators work. So they mainly work through the FRET mechanism, which is transferring of fluorescence energy from the fluorescent protein to the opsin protein. So this means that they both have to be together. So your first point of engineering would be the linker protein between your opsin and your fluorescent, the linker region between your opsin protein and your fluorescent protein. So that was the first point that we targeted. And then, in the opsin protein, what are the amino acid residues that are important for, the opsin protein is mainly a channel protein, right? So its job is to move charge from, one side of the protein to the other side. So the amino acid residues that are involved in this kind of a charge transport. So you then start targeting those positions that are important for this charge transport. So most of our improvements actually came from targeting just these two regions. And then you have the chromophore in the opsin that is going to absorb whatever light that comes out of the fluorescent protein. And there are certain residues that the chromophore would interact with. So you do get some amount of insight on which positions to target, not just based on the structure, but also on the function of different residues in either the fluorescent protein or the opsin or the region that's linking them together.
Oh yeah, please go ahead, Matt.
Yeah, no, I would also add that so the first time we found Varnam, the FRET efficiency wasn't optimal. And until then, the entire field had been mutating only the fluorophore. Nobody had actually thought about mutating the voltage sensor itself. And so one evening when Ganesh and I were actually testing the very first version of Varnam, we were wondering how we can actually improve the sensitivity. It wasn't going to work if we were mutating the fluorescent protein. So Ganesh offhandedly suggested that we should probably redshift. No, we should probably blue shift the red fluorescent protein so that there is a better overlap between the absorption of ACE and the emission of MROB3. So that it initially was kind of like shocking to me for such a bold proposition. But then when I dug up a little bit of information from the past, it seemed like there was another opsin from another species of bacterium known as Gloeobacter, where they had actually done just that. By a single mutation, they were able to change the wavelength, the emission properties of a fluorophore and of the opsin. So what we were able to do was by mutating the opsin in a particular residue, we were able to redshift it so that its absorption better aligns with the emission of MROB3. So I think that was the very
You
first
mean I
time.
said to redshift the opsin, right?
Redshift the
You said
opsin, exactly.
you said
Sorry,
I wanted to blue shift the fluorescent protein.
Oh, so no, sorry. I'm mixing things up. So I want, yes, you suggested that we redshift the opsin. So we mutate the opsin, the voltage sensor. So there was another species of opsin from another bacterial strain where, yes, a single point mutation actually redshifted the opsin's absorption. So when we did the same thing with ACE, I don't know, for some reason we found that the sensitivity actually improved, and that was a single point mutation at 81, position 81 in the opsin. So that was the first, what do I say, the first time we uncovered a way of tuning the sensitivity. And the other, when we uncovered the positive variants, that was actually a serendipitous, what do I say, serendipitous discovery, so to speak. So it was in a high throughput platform that two mutations in the ACE flipped the polarity of the responses. And we weren't even hoping to see that happen. And it was around the time that another group at Genelia, they also found a similar variant. So we were kind of, waiting to develop this idea, but then they went ahead and published it as a reversal. So they beat us to it. But nevertheless, that was a serendipitous discovery, and we hadn't strategically lined up mutations to flip the polarity. It just happened because of our screening platform. And our screening platform was actually, because of the way it was set up, it was automated. It was able to identify these positive variants as well without having any kind of algorithmic monitoring of changes. So I think there was a mix of both. In one hand, it was strategy and some literature search that helped us find these different mutations. On the other hand, it was just serendipity. It just accidentally happened when we used a high throughput approach. And I think that's why, as Ganesh mentioned, a high throughput approach is probably essential. Otherwise, it would take years for us to identify a variant that is actually going to, have an effect on voltage sensitivity. So with that,
Yeah,
yes.
that's if I get it, it's very impressive because I worked for a while as a postdoc on the Chlomeleon project in George Augustine's lab to make it more sensitive. It's a chloride indicator, and we tried many different ways, and we had also these different approaches, just analyzing the different structures with different mutations, and it's also FRET. It also uses FRET. So we use different strategies, but chloride concentrations are very low. So it's kind of, and then we try to tag it to different inhibitory receptors and so on. So yeah, it's quite an achievement. So congratulations again. And yeah, sorry for interrupting. Please go ahead.
No problem. Yeah, so that was when I kind of joined the lab. So this was after Ganesh had set up the high throughput platform. So how I came into the picture, I was working at the lab of Mike Higley, and that's where I was interested in understanding the role of interneurons. So if the audience is familiar, so there are two large populations of neurons in the brain. So you have excitatory neurons, which get activated and which activate other neurons to which they are connected. And then you have a set of neurons that are called inhibitory neurons, in the sense that they suppress the activation of their synaptic targets. So, but the problem is the two types of the two categories of neurons are not equally represented in the brain. So you have about 85% of the neurons are excitatory, whereas only 15% of the neurons are inhibitory. But then their role has been is no less important. In fact, it is some sort of abnormal dynamics in these interneurons that seem to create a lot of neurological diseases and psychiatric illnesses. So it's just that they have been understudied, but their role is equally, if not more significant than excitatory neurons. So I was interested in looking at these inhibitory neuronal dynamics at Mike Higley's lab. But then we were doing patch clamp electrophysiology, and there was no easy way to identify interneurons in slices. So pyramidal neurons, that is the excitatory neurons, had these fascinating large pyramidal cell bodies. So you can very easily identify them in slices, approach them with your pipette and, patch clamp. But then that was not so for interneurons. And the other problem that comes into the picture is interneurons are vastly heterogeneous. So there's not a single population of interneurons for you to say, okay, I've understood interneurons because I know this is how they work. Interneurons are so very many and they are very diverse. So it is important that you understand the role of each of these interneuron types. So that's how we come into the concept of subtypes. So it becomes more and more challenging to understand their role. And with these genetically encodable indicators, it becomes easier for you to identify the role of individual subtypes, because now you can target these indicators to individual subtypes that express a selective genetic marker. So, for example, there are interneurons that exclusively express a particular calcium binding protein known as parvalbumin. So if you use a parvalbumin promoter in your genetically encodable indicator, you can drive your indicator expression exclusively in the subset of parvalbumin interneurons. So that made me kind of attracted to like optical, genetically encoded optical approaches. And because I did have a background in molecular biology, it was easy for me to fit in and participate in the protein engineering aspect that was going on in Vincent's lab. And together with Ganesh's platform, it was just rigorous screening, one screening after another, and then we validated these constructs for the first time in using in utero electroporation. So we didn't even have to go all the way to make a virus and inject an animal. What we did was, as soon as we found a DNA-based variant that was having a large enough voltage sensitivity, we simply injected them in embryonic mice. We gave them brief electric pulses and delivered it into the embryonic brain. And then we sectioned the mouse's brain once the animal was born and old enough. And we looked at the fluorescence responses in neurons. So that was a kind of a quicker approach to validate these constructs early on, even before we went ahead and made viruses. So that's how I got into the entire engineering aspect as well. But then eventually my goal was to be able to use these genetically encoded indicators to look at activity among interneurons and distinct populations of interneurons. So that's how I'm actually, I mean, if you look at slide 21, that's where the story begins, where we started using our newest generation of indicators to understand voltage dynamics in genetically identified populations of neurons in the primary visual cortex. So these are all experiments in live mice. So the way we conduct these experiments is we make adeno-associated viruses out of the DNA that encodes the voltage indicator, and we inject the mouse's brain with these AAVs, adeno-associated viruses, and the mouse begins to express these indicators in the brain, and then we implant a tiny cover slip wherever we inject it in the cortex, and so that we can now look at it as if it were a slide under a microscope objective lens. So the mouse is like head fixed, so that there is no motion artifact when we do the imaging, and there is a CMOS camera that is trying to obtain pictures, and it is in the optical path. So you're collecting all of the fluorescence data right from the cortex in the mouse as it is performing different behavioral tasks. So because we are studying the primary visual cortex, we are obviously interested in how the neurons respond to visual stimuli. So one fascinating thing about the primary visual cortex is that the pyramidal neurons, the primary output neurons of the visual cortex, they exhibit an enormous degree of selectivity to features, like visual features like orientation, direction, tuning, and contrast responses, etc. So the first thing we did was to see whether this is a viable approach to see that kind of tuning in the pyramidal neurons. So what you see is an example in slide 21. You see the tuning responses of pyramidal neurons to the arrowheads, that is the drifting gratings that are going in a certain orientation. And you see that the responses are highly selective and the neurons are spiking only for gratings that are going in a certain direction and not to gratings going in the opposite direction. So that goes to say two things. First thing is the neurons are, of course, selective in their activation dynamics. And second, our approach is viable. So it is able to tweak out these selective activation patterns, and it is not giving some noise signals. So, but then the question I was trying to understand was the participation of interneuron types. So, aside from visual tuning properties, the primary visual cortex is also heavily modulated by context, which means an external cue or an internal behavioral state. For instance, depending on whether the animal is at a resting state or is awake or alert or running, the responses are heavily changing, the firing rates are heavily changing, and this kind of an increase in volume or increase in the firing rates is what is known as response gain. And it's found to be some sort of an adaptive process. Like, for instance, when an animal is stressed or when an animal is running and agitated or aroused, there is just more activity. And so my question was to understand whether interneurons play a role in this. So is there a particular way that the interneurons are maybe just not inhibiting excitatory neurons as much, and is that causing an increase in activity, or is there a different mechanism? So in slide 22, you actually see how these pathways are formed from like long distance anatomical regions. So you have two different regions that are called the basal forebrain and locus ceruleus. So these are all forming diffuse projections to the neurons in the visual cortex, and they all kind of encode stress-related signals or changes in arousal and so on. And my question was to try and understand what's going on in V1, in the primary visual cortex, when an animal is, for instance, changing from a rest state to a state of arousal. So in figure 23, this is the schematic of the experiment. So we had we injected our indicator that is packaged into an AAV virus into the mouse's visual cortex using a Cre recombinase strategy, which is a kind of a conditional expression strategy, so that the indicator was expressed exclusively in one subtype of neurons in the V1. And then the mouse was mounted on a head post, and we had access to the injected region by a cover slip, and it was allowed to run on a wheel. And we were also delivering a brief air puff that was kind of inducing a state of alertness in the mouse. And we were monitoring the pupil diameter. So traditionally, it's been found that your pupil dilates during alertness, and which is kind of intuitive as well, right? So when we are alert, we are obviously, there is an increased systemic response. So we are paying attention to whatever is being said, for instance. So we are able to track these behavioral metrics, and on the right side, you actually see how on arousal, which is on air puff delivery at point zero, at time zero, there is increased dilation of the pupil, and there is also coincidental locomotory effects. So the mouse starts running in response to the air puff. So all of these are signal a change in behavioral state. So what we were doing now is, in the next slide, you can see recording from indicator expressing neurons. So the neurons are expressing the voltage indicator, but in different subsets of neurons, one at a time. So in pyramidal neurons, in a population of interneurons that are expressing a particular protein known as VIP, another population of interneurons expressing another protein called NDNF, another population expressing SST. So these are defined populations of interneurons that are targetable by means of their selective expression of certain genes. So you're looking at spikes that are being obtained. And if you see the red dotted line, that's where the air puff is being delivered. So you can look at how spiking changes from rest and then during arousal. And you note that there are subpopulations of cells within each population that either increase their activity or suppress their activity. And because we acquire data from like hundreds of cells at a time, we can quantify these different changes. So in slide 25, you're able to see the fraction of neurons that are increasing their activity, the fraction of neurons that are facilitated versus the fraction of neurons that are suppressed in response to the air puff. And so it appeared like globally this arousal that was induced by air puff increases the spontaneous activity of multiple neuron types. So it didn't seem to have a selective neuron type effect. So our question was to understand whether our approach can tease apart the role of individual interneurons. So the nicest way we could do that was to look at interneuronal subtypes side by side. And to get that kind of side-by-side comparison, we used the duplex strategy that Ganesh introduced you to. So we tried and expressed two indicators at a time, the negative and the positive polarity indicators, to two different populations of interneurons, and simultaneously recorded from two populations. And we inferred the population identity by the polarity of the optical spike waveform. So here we use conditional expression strategies that use a Cre-recombinase driver line or a flip recombinase driver line, and we make indicators that are Cre-dependent or flip-dependent. So that Cre-dependent indicator lands in lands in a particular subset of cells that express Cre-recombinase, and the flip-dependent indicator lands in a subset of cells that are expressing the flip recombinase. So that is some sort of an intersectional strategy. So again, going back to the next slide, which is a repeat of what Ganesh showed. So you have two populations that are now expressing two different indicators, and by the virtue of the polarity of the spike form, you can identify these, identify the population itself. So what we did was went on to employ this duplex strategy in two subsets at a time. So in slide 28, you have the two variants, Ace-mNeon2 and PACE, being expressed in NDNF interneurons and VIP interneurons respectively, and we are performing duplex recordings. So what you can do in these kind of experiments is try and understand whether there is any kind of correlation between the activation dynamics of the two populations, because you're simultaneously acquiring data from them. So what the graph shows is the level of synchronous activation within each population and between the two populations. So the green plot, for instance, shows correlation coefficients between neighboring pairs of cells for the NDNF population. The blue plot shows the correlation coefficient for pairs of cells within the VIP population, and the red plot shows the correlation coefficient between pairs of cells of the two different populations, NDNF and VIP. So, and you see that there is a high degree of synchronous activation dynamics for NDNF, even more so for VIP, whereas the correlated activity is very weak between the two populations, between NDNF and VIP. And we also quantified this as a population, at the population level. That is what is seen in slide 29. So you basically don't see that the two populations having any kind of correlated activity. And we also did the same thing for different subsets of different pairs of neuronal populations for pyramidal neurons and VIP. And once again, we saw no sort of correlated activity between the two populations. Whereas if you go to slide 32, the story actually became interesting. So we expressed Ace-mNeon2 and PACE in SST interneurons and VIP interneurons simultaneously, and we performed duplex recordings in these running animals. And we found that both SST and VIP interneurons exhibited a high degree of synchronized activity. And when you looked at the correlation coefficients between the two populations, there was a strong negative correlated behavior, which means that every time an SST interneuron fires, a VIP interneuron stops firing, and every time a VIP interneuron starts firing, a neighboring SST interneuron stops firing. So that was a kind of a mutually antagonistic behavior. And this was extremely, this was another level interesting when we looked at how these dynamics change during rest state and during a state of arousal after air puff delivery. And that's the data we see in slide 33. So you see that while under normal conditions, baseline conditions, SST and VIP cells already exhibit a negatively correlated activity. When you deliver a brief air puff, the mouse changes state to arousal. This kind of negative correlation becomes even more enhanced. So there is a further increase in the negatively correlated dynamics between the two populations. So to illustrate this in the form of a video, if you can play the video in slide 34, you can actually see how the dynamics of spiking between the two populations is negatively correlated. So every time SST interneurons fire, VIP interneurons are suppressed, and vice versa. And this becomes even more prominent as the pupil dilates, which is when the mouse switches state from resting to arousal. So what is a consequence of this kind of antagonism for the circuit is something that is still a long-standing question. But then what we'd like to highlight today, the take home, is that using this duplex technology, using the two different polarity indicators, you can simultaneously observe the spiking dynamics of two subtypes of interneurons in a manner that has never been possible before. So you can actually understand the relative roles of these interneurons. So it is no longer a challenge to be able to record from identified interneurons. So hopefully the technique would enable us to see how these interneuronal activity dynamics change during normal versus disease states. And as I said, inhibition plays a very important role in a number of disorders, including neurodegenerative diseases, psychiatric illnesses like schizophrenia, neurodevelopmental disease like autism. So it might be really important to try and understand how interneuronal dynamics are altered in these situations. And I think duplex offers a powerful approach to be able to do so. So I think with that, I'm open for any questions or remarks, and then the floor is all Simon's.
Yeah, thank you so much for this. This is such interesting results that you showed here, and it's really beautiful to see how you can image really the different populations in such a way and really dissect out like how. Oh, I'm sorry. Sorry. um, I let the person up and it was a mistake. Um, so yeah, it's really beautiful to see the activity and how you can dissect out different populations. I think this will help so much in knowing how different type of populations are active in different states, and also probably in the future, like behaving animals, fear conditioning, I don't know, all kinds of. So this is really wonderful and a great tool for neuroscience. And yeah, you've alluded a little bit in the different roles of the inhibitory interneurons. It's really interesting how interneurons play such a different role at the same time point. Do you think this is a very unique characteristic of interneurons, of these interneurons? And do you expect, or would you say that this is really important for, precision of information transfer in the awake state, and because this will help us also to model maybe different artificial neural networks and so on. Thank you.
I think that's a brilliant question. And yes, that's probably where the science is headed. So, the other aspect that I didn't touch on is we also have spatial information on this kind of dynamics. So, uh, it might be that it's not only an aspect of when the interneurons are firing or suppressed, but also where or how far apart their connections lie. So to be able to model an entire microcircuit dynamics, if we have a clear picture on both the temporal and spatial aspect of inhibition, I think we will get a more complete picture of when and where these interneurons actually fire to regulate the activity of, to modulate the activity of the output neurons. So, and of course, I should also mention that with increasing transcriptomic data, we are now able to see that there are very many flavors of these interneurons. So they do not belong in a single category, and there are subpopulations and subpopulations, and it just becomes so difficult to have a unanimous understanding. So they are not of one kind, they're extremely heterogeneous, and it's probably in different behavioral context that each interneuron works, or maybe they all work, but they are more spatially or temporally differentially recruited by even the same behavioral paradigm. And I think with these indicators, you get that kind of both temporal and spatial information. Yeah.
Yeah, and I'm very curious to see in different disease models how if there is a baseline difference already, let's say autism, schizophrenia, all these different type of disease models, if there would be maybe a difference detectable already in the baseline, and maybe, yeah, this will enable us to see at a larger scale what is happening, and it's very important. So yeah, thank you. Are you planning on maybe looking at different disease models next?
Yes, I do, and but then I know that it's extremely challenging. There are a lot of great labs that are already pursuing in that direction, and we do have some technical limitations as well. We are trying to address with these indicators. They are not foolproof, so we want to be able to address their limitations before we can go into full-fledged disease studies. But definitely, that's where my interest lies. So maybe Simon's ready for his.
Yeah, for sure. Yeah, thank you, Ganesh and Madhu, for your part of the talk. So here I'm going to take it over, and we saw a lot of data around visual cortex, and I will mostly describe data acquired in the hippocampus, which is a subcortical brain areas that every mammal, human and mouse and monkeys, and they all have. It's highly important for memory, episodic memory, when you try to remember the last time you went to the beach or enjoy an ice cream. It's also one of the key brain region that is impacted in Alzheimer's disease, and essential for spatial navigation. So you can. I'm on slide 35. On slide 36, I'm going to introduce two different technologies that use voltage sensors. The one I've used from Madhu and Ganesh to VARNAM, which is the red-shifted voltage indicator. And this technology is different from the previous one that you have seen so far. This one doesn't give you single cell resolution, but this one can give you access to transmembrane electrical measurement from a specific cell type in a freely moving mice, whereas the previous data that have been shown have been all acquired in head restrained animal. And therefore, in this case, because it's fiber optic based, you can let the animal explore or live in its fully ecologically relevant behavioral repertoire and study the brain dynamics in the minimally invasive way. And what I'm showing you here is a recording that have been performed in the mouse foraging in the open space. And the recording is performed in the hippocampus. And what you see on the bottom right panel is two type of plots that show in the x-axis the time, which is in minutes. So it's a more than 1 hour long recording, which is also one of the advantage of this technology. You can record for a very long time, whereas single cell head restrained voltage recording is usually limited to a few minutes, few tens of minutes. And here you see two different plots on top of each other, and you can see an alternation of signal. From the bottom, it's highly a strong signal in the theta band, which is a frequency band that is specific to the hippocampus when any type of mammal with a hippocampus is running or walking in an environment. And when you stop walking, you go into a lower frequency state, which is known as a delta band frequency, limited below 4 Hertz. And you can see the mouse is alternating between running and resting. And that's exactly what you can pick up with this, what I would call optical EEGs, alternation of these two frequency band, which is a very nice validation of what you can now do as a electrical type of recording, but perform optically using voltage sensor. I'm not going into the details of the optical system on the left. If you have any question, feel free to ask. On the slide 37, I'm showing a comparison between the whole slew of voltage indicators that exist right now, and then one of the latest published, there you can see ours, Ace-mNeon, VARNAM, PACE, and PACER. And this plot summarizes you that as of today, the sensor that we, Madhu and Ganesh, have been able to design and come up with are the best in the world. You can see they are all ranked. In the left panel is the sensors that are negative, have a negative polarity, meaning the spike is negatively deflected down. And ACE and VARNAM are the best at picking up the change in signal due to a single action potential. On the next, just right next to it, you have the positive polarity GEVIs. And here again, you can see that PACE is by far the best at picking up a single action potential effect on the transmembrane potential. And the right double panel is the same type of plot, but now it's a train of spikes. It's especially important to select your voltage sensor based off burst in activity of spiking, because some brain region and cell type are highly bursting. The hippocampus is one brain region where you can find a lot of cells that have high spiking rate. So what you see and what I've been described is we have a set of 4 different voltage indicators of 2 different colors, 2 greens, 2 red, and for each color we have 2 different polarities. And as has been described before, you can multiplex, either combine 2 different polarities within the same color or do dual color imaging. And this is an experiment that have been done in the visual cortex from 2 different cell type, the same cell type that have been described by Madhu, but this type, instead of leveraging the dual polarity duplex system, we use the dual color. And what is nice is you can now have several dozens of neurons genetically identified, so you know exactly which cell type it is, and see, especially on the top, you can see this blinking is, you can study the type of clustering or spike timing dependencies between neurons, something that before this technology appeared couldn't be done. Then this is another set of experiment leveraging the capability of multiplexing the colors, and in this case is studying in the dorsal hippocampus of head restrained animal undergoing different behavioral state transition, resting and running. You can see the speed on the top. This is the color map, and you can see exemplar traces of 2 different class of neurons. Both of them are excitatory, but their specificity that they are both excitatory neurons from the dorsal hippocampus, but they are projecting to different cortical areas. One is the prefrontal cortex, ACC, anterior cingulate cortex, which is known to be important for cognition, motivation, planning. The other one is entorhinal cortex, and this one is a major play a major role in spatial navigation. It's the location of the renowned grid cell that give rise to the place cell. Both of them have been awarded the Nobel Prize in 2013, and those cells are essential for spatial navigation. They give you a spatial map of your environment. That's how the brain constructs this spatial map. And what is interesting, and you can directly see it from the raster plot on all the traces, is the green population, the one that projects to the entorhinal cortex, start increasing their firing rate specifically at the running period, whereas the one that projects in the entorhinal cortex seems to be affected by this state transition. In the next slide, slide number 40, you can see the quantification across many field of views and more than 150 cells. And the statistics stay the same, where you can see on the top plot in white is the speed, and you can clearly see the mass is undergoing running transition. And precisely at the transition, you see the excitatory neuron from the dorsal hippocampus that project to the entorhinal cortex increase their firing rate. And the right panel is the statistical quantification. You can see the red the red histogram is perfectly aligned with the shuffle, meaning there is no effect whatsoever, whereas the green histogram is right shifted. There is an increase in firing rate by 1.5 to 2-fold increase. So this is one example of a new type of experiment that can be done by multiplexing two different colors of voltage indicator. And now I'm going to show you another example how you can multiplex using the same color green sensor but with different polarities. And in this case, it's also in the dorsal hippocampus, but from two different classes of neuron, one excitatory, one inhibitory. And I recorded simultaneously the local field potential. So the electrical recording since 1930 in Hansberger, so almost 100 years ago, all basic and clinical science have relied on electrical recording. Maybe you are familiar with the EEG recording, ECOG recording that is performed in human. The big unknown on this type of signal recorded from the brain is that you have no idea which cell type contributed to the LFE. And so by simultaneously recording the LFE and the optical signal from a specific cell type, you can now start addressing question as to whether a given cell type will induce this given signal deflection in the LFP, and which cell type would be actually important for, let's say, a disease study. You can start narrowing down on which cell type you should study the molecular pathways and design therapeutic targets. So in this plot, you can see on the left the surgical approach and how the LFP and the cell are where they are localized, with all the, I think, up to 30, close to 30 neurons detected simultaneously in the same brain region. And on the right part, you see the time traces. On top, you have the LFP theta power. Again, as I explained before, theta is linked with increase in locomotion. You can see an increase at the end of the recording of theta power, which match with the increase in speeds. It's the other color bar on the bottom. And then all the single time traces simultaneously recorded. And now, because you have access to two types of information with voltage as compared to calcium. And first is the spiking, as we discussed before, but another type of information that is highly relevant for in neuroscience is the subsocial, which give you a lot of information regarding the synaptic input. And here the analysis goes as follows. You extract the theta band frequency from the subsocial activity in the LFP, which is some kind of ground truth canonical signal. And you look at the same exact theta frequency for each cell type, excitatory and inhibitory. So inhibitory class is the somatostatin neurons. Excitatory class are the excitatory neurons that project to the entorhinal cortex. And you look at the spiking detected from those two classes of neurons, and at which phases the spikes occurs with respect to the theta oscillation, either extracted from the LFP, which is the bottom summary polar histogram on the right, or from the cell on theta oscillation, which is the top plot. And the main insight here is that whereas each cell will tend to fire at the same phase of its own theta oscillation, as compared to the LFP, the ongoing brain tissue electrical state activity, only the excitatory neurons will be firing at a specific phase, which is 180-degree phase shifted with respect to the LFP, whereas the SST neuron interneurons doesn't seem to have any phase locking with respect to the LFP. So that's something that is very new to the field. Is now you can really start addressing some kind of multimodal approaches using electrical recording and LFP or electroencephalography, EEG ground truth, and look at simultaneously at the optical traces from 2 cell types, 3 cell types. In the slide 43, this is a quick example that you can not only do green duplex, but you can also do red duplex, because we have also 2 red voltage indicator with different polarity. And this experiment have been done in the hippocampus as well. So now, so far, you have seen that you can do dual color, green and red. You can do same color, dual polarity in green, same color, dual polarity in red. And so the next experiment is really how to combine all of them all together. And this experiment is again in the hippocampus. And in this time, we use a dual camera system to split the two the two colors of the different sensor we have. And on the green camera, we can pick up two different spiking activity. And this is the very first demonstration that you can record in the awake animal the subsocial and spiking activity from 3 different genetically identified cell types. And this is something that will never be able to be done with calcium sensor because calcium the latest generation of calcium doesn't have this dual polarity features. And I'm not even talking about electrical recording. Here we can pick up 60 different spiking cell that would be impossible with in vivo patch clamp recording that can barely do up to 8. That's probably the record, the world record. And the final slide on 45, it's a video you can see is how you can reconstruct the fully reconstruct movies. And on the top, you see the spatial representation and the blinking of all the cell and how different classes of neuron. You can see the red population that are inhibitory SST interneurons seems to be blinking somehow in synchrony and out of phase with the green and red, the two green and blue. Sorry. And the summary on slide 46, to summarize the overall presentation that Madhu, Ganesh, and myself I've given you, it's we show you that we have a suite of new GEVIs that enable single cell, single spike resolution from several dozens of neurons simultaneously in awake behaving mice. We show you the this new concept of duplex recording that can be done either with green or red duplex. And this new concept enables the simultaneous recording of voltage from 2 different cell types. Also leveraging duplex approach, we reveal fine scale mutual antagonism between SST and VIP interneurons in the primary visual cortex, and the role of different excitatory and inhibitory neurons in arousal in the hippocampus. We also show you dual color delta data, voltage emitting data that and some new insight about the different cell class contribution and projection specific excitatory neurons during behavioral state transition. Finally, we also show you how you can use this new multiplexing of voltage recording along with electrical recording approaches and better understand a type of signal that have been used in clinical and basic science from which the origin is still to date very mysterious. And finally, the suite of GEVI are all mutually compatible for concurrent recording of up to 4 different genetically targeted cell type. The idea being that by leveraging all those sensors simultaneously, you can have a much better understanding of circuit dynamics in behaving animals. So all this work, the last slide is about acknowledging all the people involved in this work. Vince Pierban from Yale, mentors of Madhu and Ganesh, Mark Schitzer that have been my mentor and also very helpful in this the last stretch of this project. All the different co-authors involved in this project and the funding agencies. Thank you.
Well, thank you so much for presenting this very impressive work. And it's really, this is so helpful because for example, when I was a PhD student, I used to do wholesale recordings in Joseph Ledoux's lab, and I had the colleague that was doing field recordings, and sometimes we had completely opposite results in the same behavioral or in the same, from the same animal. Like we shared slices and we had opposite results, and was always okay. It's probably in field recording you just have the whole population, but it would have been a way more easier, more efficient way to do things than to patch clamp each cell type. So this is so helpful, and it will scale up our data acquisition by such a impressive rate. So this is totally will change, how much we know about what the different cell types do and what type of behavior, and it will be way more accurate. So do you think that the future papers that come out using this will contradict some of the knowledge we currently believe to have?
Yeah, I mean, it's remains to be seen, but I'm sure it will open up new venues on a new type of experimental question neuroscientists can now address. And I wouldn't be surprised if some of the dogmas we hold in the field might be contradicted. But this remains to be seen, of course.
Yes, of course, but yeah, I'm just I'm looking, especially in the hippocampus, I'm really looking forward to learn more. Are you currently working maybe on a, biological question project right now using this that kind of can give us already an insight to, what will change in our view of the hippocampus maybe?
Yeah, so I'm actually working specifically on the extension of this technology, which is more related to the fiber optic approach that gives you access to a free behaving animal. And so I'm working with a colleague in the lab and expand this fiber optic approach to a dual color system. So you can track in the freely behaving animal from any brain region up to 4 different cell type, 2 per fiber optic if you do multiple fiber optics simultaneously, and give you really the idea of how different cell type functionally interact in the freely behaving animal. And the second technology is studying the spatial temporal pattern of those dynamics from 40 different cortical areas simultaneously. So you can see a lot of traveling waves from specific cell type going different brain region during different behavioral tasks. And I think those two also emerging technology would be something extremely exciting for the community. But more
Yeah.
specifically to the spiking, I think an interesting question would be related to memory and how different, especially because when you think you have a memory recollection, this usually happens very, very fast in the split second. And no other technology can give you access to a millisecond timescale neurodynamic as the memory is retrieved or is recollected. And so that's something that I'm personally interested.
Yeah, it's amazing. Also, developmental research. I'm not sure how well it works in the developing animal, but if it would, that would give us a lot of insight also, especially then later on in disease model.
Mhm.
How integration of different neurons maybe doesn't work very well, let's say in autism and so on. And then, addictive state, all kinds of questions could be that I always had could be actually finally addressed, which is really exciting. And then the other thing, are you planning on maybe then in the future adding on technology to maybe selectively silence different neuron subpopulations to then see what happens if you take them out, basically, very specifically?
Yeah, I think Madhu might have something. Madhu?
Yeah. Oh, yes, because we do have these red-shifted indicators, and they can be multiplexed with blue-shifted channel rhodopsins, if that's your question. We can optically activate and silence using pairs of, read-write tools. So that's definitely a possibility. And yes, I also think there's also a lot of scope to combine them with gene perturbation technologies like targeted gene manipulation and maybe even chemigenetic silencing. So there's definitely a lot of ways we can extrapolate from these indicators. But then right now, the major puzzle is to be able to target the 4 indicators to 4 different cell types. So that's kind of the only pressing challenge. We've been able to target them to 3 non-overlapping cell types, but the question is, how do we target them to 4 different cell types without overlap with one another? So that's one limitation we will have to try and address. And that's because of an existing limitation of genetic targeting approaches and mice lines with these driver recombinases. And the other, one more limitation of our indicators is that they are also subject to photo bleaching restraints. So we ideally cannot do continuous imaging for a very long time. They are subject to photo bleaching, which kind of reduces the SNR, the signal to noise ratio. But we're also hoping to screen for more photostable sensors or use fluorescent proteins with greater photostabilities to try and improve them in that regard. Does Ganesh have anything to add?
Yeah, I mean there are very many things we can do with these indicators that were not possible before, but there's still long ways to go. Um, so these are just kind of scratching the surface of what we can do with these indicators to try to understand even how different populations of neurons interact together, even in the superficial layers of the brain. But if you want to go deeper into the brain and understand from even more populations, we have a lot more work to do in terms of both improving the indicators and also having better imaging techniques that are more suited for these kind of indicators. But this is already kind of pushing the boundaries of what was possible just a few years ago. As I showed, even in voltage indicators, it was only 5 or 6 years ago that we were just able to record them in vitro in cultured neurons or in slices. And now we are thinking about triple population in voltage imaging in awake behaving animals. So that's a giant leap, but yeah, more work is to be done.
Oh no, this is very impressive, and it's a giant leap. And if you add, let's say, different neuromodulators, agonists, antagonists, and so on to these experiments, there's so much to be learned with 3 populations. So yeah. But Simon, did you want to add something?
Actually, your last thought will open up my own comments. So we focus today on voltage, but the field of genetically encoded fluorescent indicator, so much broader, which now include not only GCAP and voltage, but also all the neuromodulatory sensors. You have dopamine, norepinephrine, acetylcholine. Some neuropeptide now are available, and they're all mostly green, and very few maybe dopamine sensor is red. So you can even think of studying the role of neuromodulation into the cell voltage activity by combining different colors of between voltage and neuromodulatory sensor.
Yeah, I was thinking about when I went to a conference, but now I forgot the name of the person I presented. That in the very beginning, we gained insights, like crucial insights, with like the beginning of neuroimaging, how acetylcholine works in the cortex a little bit better. But this will give us way more precise answers to how drugs and neuromodulators work and what they are changing in the network just by pouring it on there. I mean, just just pour different stuff on the slides, and you will gain a lot of insights, and then compare it to different disease models that will add up even more. But we have to have probably enough fun, like funding people doing it. But this will reduce the cost of gaining those insights by a scale of thousands. Because if you imagine, before we had patch clamp recordings only, basically, and then we started having some neuroimaging, but still not at this precise level. And how many cells can you get a day? Like, if you're really good, maybe 10 cells a day, and you have to pay one person to do that. And now you can have 3 populations in one slice. This is like such scaling up of efficiency, which I think will help a lot the field.
I agree with you on that. And also experimental mice. So you're studying multiple populations in the same mouse. So you're minimizing the number of mice you need to sacrifice. So that's another, conservation.
Yeah, and if you have, in one postdoc I had to cross indicator mice with disease models. So since they were in a different genetic background, you had to kind of double cross them. So, we had very little outcome of mice we could actually use. And then you get very nervous on not
Yeah.
wasting them. So, for these type of experiments, it's also, very, very helpful. So,
Yes, they'll be normalized for the same background. So, whatever the background is, it's going to be the same. So you're looking at multiple populations in a defined background. So there's little variability from the genotype of the mouse. So that's kind of an added advantage.
yeah. And then if you use virus and then you try to compare different different populations, that's really not good, because it could just be you injected a little bit more than in the other, and it messes up your data. So that's another issue with using virus injection to express these. So
And also in terms of cost effectiveness, I think one aspect which can also be a little bit of a downside is the fact that we don't need a 2-photon microscope. So we do our recordings with wide field epifluorescence imaging, and the downside is that we cannot image very deep in the tissue, but then there are ways to go around it. But the advantage is you don't end up spending $500,000 in a 2-photon setup. So it's extremely easy to reproduce and to set this up in a lab. And even even with the dual polarity concept, you literally need just one channel to record from 2 populations. So that again minimizes the need for more sophisticated instrumentation, cutting down costs.
yeah, definitely. Yeah, this has so many advantages, and it's such a wonderful work. And I know you probably have to go, because I think we went way over the time we agreed on. But this is this is so interesting, and it's really very helpful work for the field. And congratulations, and I really wish all the best for the future, and keep developing these technologies. And it's such a great contribution. And
Yeah,
yeah,
I
I
think
hope
that
all the best.
there was just one question from someone on the chat. The very last
Oh, I
question.
see.
Yeah, someone asked if we ever use these voltage sensitive opt-ins along with the what we discussed before. Just to answer it for him, yes, we did use these indicators with the channeled options for stimulation, because the red voltage indicators, we've combined them from with blue shifted channeled options to stimulate cells and record from them. at the same time. Okay, sorry.
Yeah, I guess we were kind of concluding, and I also wanted to take a moment to
Yeah,
thank Katerina and, yes, the entire team at Science Society for choosing our paper to discuss. And yeah, this has been a collaborative work, as I said, and we've been happy to have such a wonderful team. Ganesh and Simon, and Simon's colleagues as well, Cheng, who's not on the call today, as well as the professors, Vincent Perbone and Mark Schnitzer. So yeah, this has been really, really exciting and challenging for the field.
yeah, and we're
Yeah.
also more excited about what new things we can do with this technique, and also getting others up to speed on using these voltage indicators and dual polarity multiplexed imaging. We are very big on sharing these things openly to the community. So we share our voltage indicators freely to the neuroscience community, and they can get these indicators on AdGene, or even reach out to us via email, and we'd be happy to share these indicators either as plasmids or viruses that you can directly inject in mice, and also help you with setting up voltage imaging or do data analysis. So if anyone is interested in the technique, please feel free to reach out to us. And also shamelessly, because Madhu and I, we both just recently started our group, we're also looking for people to join the loop, to join the lab, to do more of these kind of voltage imaging experiments. So if you're interested in doing that, please also feel free to reach out to us. And we definitely have open positions in the lab for people interested. And finally, thank you, Katerina, for having us.
Yeah, thank you so much. No, it was really an honor and pleasure. And yeah, feel also free to reach out to me if you don't know the contact. I will, connect you with people, and I hope you get really great people that join your lab, because it's really wonderful work, and I think people will learn a lot in your labs. So good luck for everything, and I wish you all the grants and and smart people. And
Thank you.
yeah, thank you so much. And maybe once you have some of the future work that we discussed a little bit, you could come back maybe next year or so. That would be really wonderful too. We will definitely
That'll be
follow your
awesome. Yes.
Absolutely.
yeah.
Definitely.
Thank you so much. And yeah, thank you everyone for coming, for asking in the chat and commenting. And if you like discussions like this, our club will have the next talk next week with Dr. Illich, and he developed a way of moving objects with ultrasound waves. So if you feel like reminded of Star Wars, I was when I read it, but it will be an interesting talk. Very different, but yeah, also interesting. So thank you so much, and enjoy the rest of your day. It was such an honor having you. I really appreciate it. And yeah, good luck for everything.
Yeah.
Thank
We
you
will.
so,
Thank you,
so much.
Katarina. Thank you
Thank
all.
you.
You
Okay,
have a good day. Bye.
Thank you. Bye-bye.
thanks. You too. Bye. Close the
Bye.
room in 3, 2, 1. Bye, everyone. Thank you.